Today, we are going to talk about somatic embryogenesis, the direct and indirect method of somatic embryogenesis, and the factors that affect the somatic embryogenesis. So, before going into the class, shall look into a bit of introduction what we were speaking throughout our previous videos. Students, indeed, you have to appreciate tissue culture because now you can appreciate the large number of cells that can be produced from a small explant, which can give rise to callus, which can be multiplied indefinitely. So, once a culture is initiated, that becomes a gene bank. So, whenever you want to grow a cultivar which is not available, an unique cultivar with an unique trait, you can always go to that particular gene bank and retrieve the plant or your somatic embryogenesis or the gene plasma which is present, and you can multiply these cells anytime, and these cells can be subjected to different conditions, different media conditions, cultural conditions. So, this goes on to show cells can grow, tissues and organs can grow into a whole plant. And why this happens? This all happens because of the totipotency ability, the ability of the cells, tissues, organs to form callus suspension cultures. Everything happens because they have the ability to grow into a whole plant. And this phenomena is what we told it as totipotency. So our scientists have used this terminology or the phenomena of totipotency and have applied it into a wide variety of crop plants and agricultural plants for their improvement. So throughout our previous videos also you saw that how these are being produced, your organ culture, your ovary, ovules, meristem, micropropagation, somoclonal variation, and today you'll be studying about somatic embryogenesis. So the scientists have developed embryo from somatic cells. This is all is possible because of totipotency. And you agree that with me, right? Okay. So now we shall go into and see the definition. What is this somatic embryogenesis is, and how can we develop this embryo into a whole plant? So here you can see the pictures. These are all the protocoms which can be developed into the shoot and into the root. And finally, you can see that how a new plant is being produced. So before going into the details of it, first let us define what is somatic embryogenesis is. So, somatic cells are any cells of the body except the germline. Germlines are what? They are the egg and the sperm that is formed by the sexual reproduction. They form the gametes and then they form the zygotic embryo. Leaving apart these germ cells, other all cells are the somatic cells. And what is the meaning of the word embryogenesis? Embryogenesis is the normal formation of embryo. So, somatic embryogenesis means I am going to develop an embryo from the somatic cells, that is a non germ line cells. So, is it possible? And you can see that the answer is yes, it is possible. So, you can see that the somatic embryogenesis, okay, this is pictures, you can see that it is a formation of adventitious embryos, the production of embryo from the somatic or non-germ cells. So you can produce somatic embryogenesis by two methods. Okay, this one I'll be explaining the next slide. See, the two methods are the direct embryogenesis and another is the indirect embryogenesis. So, in direct embryogenesis, here the somatic cells get converted directly into somatic embryogenesis without the intervention of the callus. That is, callus is not being formed. I can take the explant, okay, and directly initiate the formation of the somatic embryos without the formation of the callus. That is known as the direct embryogenesis. So, what happens here in direct embryogenesis? The cell, okay, the cell itself is getting transformed into somatic embryos. So the plant produced from these embryos will be the mirror image or exactly or identically same to the mother plant. So it is ideal for micropropagation. Is it right? Obviously, because you have taken explant. What is an explant? We told it is a part or a piece of a tissue or a cells or the organs from a plant what you have chosen. You have chosen that explant and you're directly developing the embryo. And that embryo will develop into a whole plant. When it develops into the whole plant, don't you think so that it will be exactly the same as the parent plant or the mother plant? This is what we said even about vegetative propagations. Wherein the previously you studied about the adventitious micropropagations, right? And axillary bud propagations. This all led to what exactly we are getting the clonals of the parent plant. So micropropagation, whenever you want to do micropropagation, that is, if one plant, for example, say this, uh, let me draw and explain to you all. Okay. For example, this is a plant. Okay, from this is a plant. From this, I took the explant, and this explant, what happened? It produced the embryos. So this embryo will develop into an again a whole plant, which will be same as the mother plant. So it is nothing but you can use it for micropropagation. If this plant is having a unique property, okay, which you are interested in, you can grow that plant, produce the stomatic embryos, and produce the same variety of plant. And this phenomenon is known as micropropagation. So, what actually is leading to the development of the egg plant directly into the somatic embryos formation is there are what are called as pre-embryonic determined cells. Here it is, pre-embryonic determined cells. So, these cells, okay, they are present in the somatic cells, and these cells are what they are committed to form embryos. Okay, so since they are committed only to form embryos, these can form the somatic embryos. For example, embryogenic tissue. If you take an embryogenic tissue as an explant, this embryogenic tissue is already destinated, or it is committed that it can form only the embryo and nothing else. Okay, that is called as pre-embryogenic determined cells. So, for example, is the embryogenic tissue. You took embryogenic tissue, which is a somatic, you wanted to develop a somatic embryo, and obviously it will produce your somatic embryos because it is already committed to form the embryonic cells. Another method is indirect method. Okay. So in indirect method, as you studied in organogenesis, what here you can see, you take the explant and the explant produces here the callus. From that callus, you are able to produce the embryos. So here what happens, one extra point you have to remember, or an extra step is there, that is explant, callus, and then the embryos. So now this is called as indirect embryogenesis. So when, for example, if I take the explant from this, okay, from this plant which is having unique characteristics, I am developing callus. Okay, I am adding all the media, all the requirement ambient conditions I am providing. And you have studied in our last class about somoclonal variation. Okay, that in the somatic cells there can occur variation due to various factors. Maybe it is stress or the growth regulators or the environmental condition can lead to the stress. Then what happens? It leads to variation. So there is always a chance whenever I develop callus, I will not get the mirror image of my plant, my mother plant. It can be different from my mother plant. And that is what is the variation, because it would have undergone mutation. So if that mutation variety is useful for you, you propagate it. You micropropagate that. If it is of no use to you, just discard it. So this is the direct embryogenesis and indirect embryogenesis. Now in indirect, okay, what happens? You take a explant, you develop a callus. Okay, that is, it has to what are callus? Undifferentiated mass of tissue. Then you provide the media wherein the dedifferentiation occurs, and this is known as induced embryogenic determinant cells. These were already predetermined. They were committed to form embryos. These were induced to form embryos. That is, you had a callus. You could have no need of you could have chosen your own way. Either you could have grown the callus into a whole plant, or you could have grown it into a suspension culture. But what is your target? Your target was to develop the embryos. So you induced embryogenic in the callus. Hence it is called as induced embryogenic determined cells. Right? So in this step, okay, the explant first is produced from an undifferentiated cells. Who are your undifferentiated cells? These are your callus, which is then differentiated into somatic embryogenesis. So the callus may show variability, variation, and if our interest is in the variation, we can induce a particular variation in that callus and we can propagate that plant vegetatively. But there are some drawbacks in indirect embryogenesis. What are these drawbacks is, the problem with the callus is that it divides very fast, and not all the time the chromosome duplication occurs. It is not so that every time there can occur duplication of the chromosomes. No. Therefore, a callus will have cells with different ploidy level. Ploidy means the number of chromosome, either that is the set of chromosome, like diploid, haploid, tetraploidy, triploidy. It can be any type of ploidy. Okay, so the level with that means these callus can have or they produce the ploidy levels as well as different kinds of chromosome. The chromosome may be different from that of the mother chromosome, mother plant chromosome. So you can say that this callus cell, the chromosome would have duplicated, it would have duplicated twice, it would have duplicated thrice, or it wouldn't have duplicated at all. So we can get different kinds of plants from the callus. Either you can get a somoclonal variation or you can produce even a somatic embryogenesis can be produced. Only thing here is the variation can occur. Whereas here in direct embryogenesis, there is no question of variation. You will get a plant that, I mean, the plant will produce the same characteristic that of the mother plant. That is the it is a mirror image of the mother plant. So here what you can do is you can go for only micro propagation. Same clones of plants I can provide here. Because of the formation of the callus, I can do with some manipulation in the callus and develop some variation. And the callus itself is not stable. Okay, it can duplicate, the chromosomes can change in them. So that is about the indirect method of embryogenesis. So students, whenever we are going for somatic embryogenesis or any other type of tissue culture, the first point what you have to remember is before subjecting the plant to de-differentiation, somatic embryogenesis involves thorough selection of the explant, careful selection of the media, plant growth regulators, and the physical environment in which your plant is growing, that is the growth room, culture room, the incubation time, the physical parameters like light, temperature, density, all this matters. Okay, simply we cannot tell that direct embryogenesis and indirect embryogenesis is formed. No, these points you have to remember. You have to go for a first see that you screen their plants, and after screening select the explant, particularly the shoot tip and the mature embryo and the young floral plants are used. So this involves a series of process for stimulation of somatic embryogenic cell. So there is a series of process that stimulates the somatic embryogenesis. There is transformation through germination into a complete plant. So what happens is I have taken my explant, I will stimulate it to develop into the somatic embryos. After somatic embryos are formed, what will I do? I should try to transformate it, tell or instruct them to germinate into a complete plant. So these points should be kept in mind when we are doing any type of plant tissue culture. So here you can see the picture of your somatic cells. Why we have chosen somatic cells? Because our entire idea or the aim or the scope of our experiment, what we are going to study is the somatic cells. I am taking the somatic cells and either by direct or indirect method, I am going to develop it into a somatic embryos. So students, The process of formation of embryo is called as embryogenesis. Embryogenesis starts from a single embryogenic cell. That can be a zygote. What is a zygote? It is a product of the fusion of an egg and a sperm during the fermentation, or an undifferentiated callus cell. So, zygote, it is naturally formed. Okay, but is the callus cell naturally formed? No, from callus cell we are developing the embryos. So the embryo developing from the zygote are called as zygotic embryos, while those derived from the somatic cells are called as somatic embryos. So during embryonic development, the polar axis of your plant is established. The domains that set up the organization of the plant body is D-lined. Okay, what I mean to tell is D-line means, one second, we use this word D-lined. That is to say that you are committed to form a particular thing and nothing else. Okay, it is designated to do that. So during embryogenic development, polar axis of the plant is established, and the domains that set up the organization of the plant bodies are defined. That is, they are committed, they have to know what they should produce. Okay, it is well defined within the cells. I should produce a somatic embryos, or leaves, or stems, or floral parts. Okay, this is already being dictated to the plants. In in the nature also, you can see that it is de novo which is happening there also. So the primary tissues and the organ system, as I said, are D-lined. So here you can see that we have taken the somatic embryos. The embryo development occurs through the exceptionally organized sequence of cell division. So your embryo formation occurs through an organized sequence of cell division. Okay, your cell has to divide, it should undergo enlargement, and it has to undergo differentiation. Remember, students, your somatic cells, whatever you have chosen, okay, it should be organized sequence of cell division. However it want, it cannot divide. Okay, it should be a well organized sequence of cell division. It should undergo enlargement and differentiation. Whether it is your zygotic or your somatic embryos, share the same gross pattern. This is the same pattern they follow, whether it is your somatic embryos or it is your natural zygotic embryos. So embryo development is always bipolar, having a shoot pole and a radicular pole at the opposite ends, right? So always, whether it is your zygotic or somatic cells, they are always bipolar. You have a shoot pole at one corner. In the north pole, you have your shoot, and to the south pole, you have your root. So these are both opposite ends. So embryos are not organs. Remember that. Embryos are not organs because they are structurally independent from their parent body. So this is your somatic cells, and when they develop into your somatic embryos, do you think they are attached to the parents? They would will be in the beginning, but then they will detach from their parent, and it is no more a part of that plant. So you can't call it as an organ. That is, they do not have the vascular system connection with the parent plant body, no more. Okay, so once they develop into a embryo with the shoot and the root poles being formed, they get detached from their parents. That is, no more they'll have a vascular connection with the parents, and so you cannot tell these embryos are organs of that plant. Now these are the independent embryos which can develop into the plants. So you have, see, this is a somatic cells. So what is, in other word, I can tell this is a progenitor cells. And these progenitor cells, no, that is your starting material of the cells. These always undergo unequal division. So the physical observable transition from a non-embryogenic cell to an embryogenic cell in the somatic embryogenesis appears to occur when your progenitor cell undergoes an unequal division. So when this unequal division happens, what it results? It results in a larger vacuole. Okay, so there is a large vacuole in the cell and a small densely cytoplasmic, that is the embryogenic cells. So the embryogenic cells then either continue to divide irregularly to form a proembryonal complex. So they are keep on dividing, and when they divide irregularly, these are called as proembryonal complex, or they divide in a highly organized manner to form what? Somatic embryos. So my somatic cells are known as the progenitor cells. They undergo unequal division. Okay, and this division, this is a, whatever is happening, it is a physical parameter which is occurring. So the physical observable transition. Okay, so what is transition? Transition is that from somatic cell they are being transformed into an embryo. That is known as a observable transition from a non-embryogenic cell to an embryogenic cell in somatic embryogenesis appears to occur when your progenitor cells undergoes an unequal division, resulting in a large vacuole with a dense cytoplasm. So the embryonic cells, then what they do is they either divide irregularly to form proembryonal complex, or they divide in a highly organized manner to form a somatic embryo. So this is gives you a clear idea or the picture what is your somatic cells. So you can see that how the somatic cells are undergoing various changes. Okay, see, cell division occurs, vacuolated cell, then you can see here embryogenic cells. And what happens? Your embryogenic cells are dividing. They form the apical cell and the basal cell. Whereas you can see the vacuolated cell. Okay, they are enlarging themselves, and this enlargement part is known as the suspensor. And what is happening here? Your embryonic cells, they from form two cell embryos, four cell embryos, and they can finally they become a quadrant stage, and wherein they become bipolar. That is, they'll have an opposite axis of developing shoot and the root. So whenever we take a cell, and if our idea is to develop it into a somatic embryo, of course, we have certain stages what we have to follow. And the first stage of the formation of your embryogenesis is, as you have seen here, okay, induction, then maturation. then go for germination. So these are the various steps which are required for the cells to undergo the embryo. Look at this point. The bipolar structure with close radicular end rather than monopolar structure. So we don't find a monopolar structure in the embryo. Instead, we find a bipolar structure to form the shoot and the radicle. The embryo arises from a single cell and has no vascular connection, important point, with the mother tissue. Okay, this point you have to remember. That is, what happens is they do not have a vascular system connecting them with their parent plant body, and hence I told that they are not the organ of the plant. Indeed, these are embryos which are going to develop into a new plant, hence they have no vascular connection. So what is the first step in the formation of embryogenesis? It is known as induction. I'll explain to you all like how induction all this happens in the next slide. But do remember these. It is the initiative phase or the starting phase where your somatic cells are induced to divide and differentiate into a group of meristematic cells. Obviously, if your cells have to divide and differentiate, you require meristematic cells. Okay, and they divide, and what they form an embryogenic clumps. Next, what comes? Maturation. Okay, you have induced the formation of the embryo. You have initiated, but it should mature, right? So, in maturation, in this phase, your somatic embryos develop into mature embryos by differentiating, and the mature embryo here undergo biochemical changes to acquire hardnesses. Okay, maturation is certain changes which is happening in the somatic embryos so that they harden themselves. How they harden themselves also we shall look. So what is happening? The body of the embryo is preparing itself to undergo maturation. That is, the biochemical changes is happening in the cells. So for these two, like induction and maturation, what are the factors that are affecting your somatic embryogenesis? We shall look into that. See, for the induction, okay, for the inducing your somatic embryogenesis, you require growth regulators. So the presence of auxins, that is 2,4-D, is an example of an auxin. So your 2,4-D chlorophenoxy acetic acid, or simply called as 2,4-D in the medium, is essential for induction of the embryogenic clumps, and maturation is achieved by culturing the somatic embryo on a media containing some doses of cytokinin and high sucrose and abscisic acid. So this point you have to remember. When you're going to induce an embryogenesis formation, you have to provide auxin. And what type of auxin? It is the 2,4-D auxin. After that, you have induced your somatic embryo, it has to undergo maturation. When it has to undergo maturation, herein you are going to provide a different regulator which is known as the cytokinin, and also you can provide high sucrose, and also abscisic acid also is provided. What is abscisic acid? It is also a growth regulator. But also it is being found, if you add a combination of cytokinin, example benzyl aminopurine, and auxin like indole acetic acid and gibberellic acid, is shown to have a good progress in the somatic embryogenesis formation. Sometimes Silver nitrate is added as an inhibitor of ethylene. So ethylene is also an gaseous hormone. So what happens, this ethylene can interfere with your somatic embryogenesis formation. So therefore, to inhibit this ethylene, I can add the silver nitrate. And high sucrose in the medium is required for maturation of your somatic embryogenesis. And not only maturation, it is also required for your steady and hardinesses of your embryo. You heard that word in the previous slide in the maturization. Okay. So, nitrogen, how you're going to add this nitrogen to your medium? You can add it in the form of ammonium. Nitrogen sources like ammonia can be added, okay, for the induction of the somatic embryogenesis. So, you can add ammonium nitrate or ammonium phosphate. So, this ammonium will be absorbed by your plant. So it is very essential for induction of somatic embryogenesis. Also, you can use nitrate. Look at your nitrate is required during the maturation phase. So nitrogen is a gas. I can't add a gas to my plant. So instead of that, I will add in a combination, that is ammonium. You know that the salts of ammonia contain nitrogen. You add it to the media. So what, your plants are going to get nitrogen from that. And afterwards you add nitrate. Okay, you have potassium nitrate. An example I'm telling. The plants will use up the nitrate from that and it will undergo maturation. Then the genotype of the explant may also influence the potential of regeneration of your embryogenesis. What is the genotype of your explant you have chosen? That is also very important. Also, you can go for a high potassium level and dissolved oxygen level to promote somatic embryogenesis. So also polyamines. So you can see that throughout the induction step, we have seen that different concentration of synthetic auxins, especially 2,4-D, is used for callousing induction. It is, and its concentration also varies. Usually, no, like we add around 0.5 to 1 milligram. Okay, 1 milligram per liter we add. What we add, it is the 2,4-D. So it is induced in the callus formation and the concentration is usually zero point 0.5 to 1 milligram per liter, which is good for the callus induction and proliferation. And auxin supplemented media to induce embryogenesis, it must be transferred to the cytokinin media. Okay, you have induced your somatic embryo. Now what, it has to get matured. Okay, it has to undergo development. So what you are going to do is you transfer it into a cytokinin media. Your cytokinin can be what, it can be a zeaxanthin, but I told you that BAP is more commonly used. Okay, it is most commonly used in this combination. BAP, IAA and gibberellic acid is very good. And pH modification also we can do. We can go for high sucrose in the medium. Okay, high sucrose concentration without growth regulators have also found a place in the embryogenesis. Nitrogen requirement is also essential for the induction of the embryo formation, and it is required for the induction as well as the maturation of the embryo. So you can add your ammonium chloride or ammonium nitrate, casein hydrolate. All range of amino acids have been used for the induction of the embryogenesis. And one more point what you have to remember is like oh, it is usually observed particularly you see that most of the experiments are being carried out in the carrot plant. Okay, so it has also been observed that the initial cell density, what is the density of the cell, does it play a role? Of course, even the cell density also plays a role. Density. Okay, your cell density also plays an important role. So, it is a critical factor for differentiation of somatic embryogenesis. In carrot plant, it is observed that high density culture on oxen-free media resulted in the inhibition or the formation of your somatic embryogenesis. So what we are telling in for the example in carrot, what they did was the cell density was more and it was an oxen-free media. What did it happen? It resulted in the inhibition of the somatic embryogenesis formation. This is because the reason why it inhibited is that the cells which are cultured, okay, there are cells, carrot cells are present, and these cells at high density have the tendency to release high amount of phenolic compounds. We have already spoken about phenolic compounds, and you know that they can act as a inhibitors, right? So your high cell density can provide a high amount of phenolic compound which can inhibit the formation of the embryogenesis. But remember students that there is also a minimum density requirement because embryogenesis cells also releases certain promoters also. Okay, it is not that they only inhibit in I mean they produce inhibitors. There are that a particular cell density is required and that should be standardized for each plant because cell density is important. If it is very high as it is observed in carrot, it will act as an inhibitor. But if you're adding the correct amount of cell density, it can be useful because it can also act as a promoter. And also, I told you that pH. In some plant, what it has been observed is the pH modification and high sucrose concentration without growth regulators have also found a place. So, coming to our development stage, whenever you are in the development stage of your embryo, you should avoid auxin. Otherwise, the auxin can inhibit the embryogenesis. And your the different stages of your embryo formation, somatic embryogenic formation, is very much similar to your zygotic embryogenesis. There is no differentiation. See, it forms a globular, heart, torpedo, cotyledonary, germination. These are the stages of your development of your embryo. So we shall look into the various stages of this embryogenesis. Look at this. You should have a blueprint in your mind about the structure. Okay, there's different stages. This is a very important question. So what is the first stage? What is the name? It is the globular. Somatic embryos generally originated from a single cell and they develop into a somatic embryo. That means what I mean to tell is don't get confused. Somatic cells, you are converting them into somatic embryos. Until now, whatever you saw, that is the growth regulators what you have to provide so that they undergo induction, they undergo maturation, now they have to undergo development. So and the first stage is the in the somatic embryos arises from a single cell which divides to form a group of meristematic cells, and these cells become isolated by breaking the cytoplasmic connection with the surrounding cells around them. And subsequently, after they have broken down all their connections, okay, cytoplasmic or vascular connections, what they do is they undergo cutinization. Cutin, CUTI, cutinization of the outer walls of the differentiating cell. So this cell is having medicine, so it is differentiating. So what it will do is it will isolate itself from the other cells. Okay, it breaks the cytoplasmic connection, and then it will develop a cutinization in the outer walls of the differentiating cell. It increases in size by further division because it is full of meristematic cells, and it forms a spherical shaped structure which is known as the globular stage. At this stage, the primary meristem, that is your protoderm, your procambium, will all become visible. Okay, so what is the meaning of this? The procambium and all, you know. Okay, this is protoderm. Okay, your protoderm and your procambium will all be visible in the globular stage. So protoderm is what? It is an outer layer of the meristem in the embryo and growing point of the roots and stem. So this is the growing point of your root and stem. They are the part of your meristems. Sorry, what I'm writing? Root and shoot. Okay, shoot. SHOOT, shoot. So protoderm is the outer layer of your meristem in the embryos that are growing point of your shoot and root. In the second stage is the heart-shaped stage. This is the next stage in which the meristem mass of cells continue to divide and differentiate with the introduction of cotyledons, and that is the cotyledons primordia is formed. What is primordia? I told it is the earliest stage of the development. Okay, so here a primordia is formed, and what it forms is it forms the cotyledons, and it transforms into an heart shaped embryo. So here the cotyledons are readily formed. They undergo multiplication and they form cotyledons. Then comes the third stage. And the third stage is the tropidot stage. That is, the embryo grow further, developing the cotyledons and attains the torpedo shape structure. And the last one is the cotyledonary stage. Okay, the growth of the embryo in this involves the division of the cells inside the cotyledons ring to form shoot and the root apical meristem. So here what happens? The division occurs in the cotyledons to form your shoot. Okay, shoot and root meristems. And the differentiation of your procambium also occurs, and this takes place leading to the production of a bipolar structure. These are bipolar structures. So this is what you saw in the other picture also, right? So finally what happens? A cotyledonary stage is produced, wherein it involves the division of the cells inside the cotyledons ring to form what? Shoot and the root apical meristem, and there is differentiation of procambrium takes place, leading to the production of a bipolar structure containing the root and the shoot axis. So these are the four stages what I explain now through that picture that is here. So you can just have a glance into this. So the first one is the globular stage. Primary meristem, protoderm, and procambium will become visible. That is, they are forming the point, growing point of your root and the shoot. Secondary stage is your heart-shaped formation. Cotyledons are being produced. Next is the torpedo shape. Here the embryos further develop into the cotyledons and differentiation occurs. And finally here you can see that the radical or the plumule, that is the root and the shoot, are being produced. These are the four stages of the development of your embryo, whether it is zygotic embryo or somatic embryo, it is one and the same. So, apart from these, all what we have spoken, what else can influence the formation of your embryos? One is the epigenetics. Okay, epigenetics controlling the expression of the gene has proved that auxin, in addition to the modification of of the DNA methylation pattern, influences the embryonic cells, and also the other factors that regulate are the proteins, your post-translation modification, protein turnover, protein-protein interaction. There are many factors which are involved. Okay, that is the molecular biology part, like your post translation modification, what is the protein turnover, what is the interaction of the protein protein, all this happens. So epigenetic controlling the expression of the gene has proved that auxin, in addition to modification of DNA methylation, so you have the, I mean, the plant have their own DNA and they are undergoing modification. In addition to that, what is present? Auxin is present. Okay, and addition to that, what is present is the epigenetic controlling the expression of the genes. So, what is this epigenetics? Why it is controlling the genes? What do you mean by that? So, epigenetics is the studying of how your behavior and environment can cause change and affect the way you grow. That is known as the epigenetics. So already the DNA, okay, it is bringing about certain modification in your embryogenic cells along with the auxins, and also along with this the epigenetics, that is how your behavior with that of the environment can bring about some changes that affect the way you, I mean, the plant grows. And always remember that epigenetic changes which influences the plant is always can be reversible. Okay, that is, you change the environment, then it will be different, but these will be the same. Okay, the DNA modification, all that, because these are the molecular, at molecular level, the variation which is occurring. So the embryogenic development changes in expression occurs, molecular markers indicating the transition have been recognized, and also the gene that enhances the embryogenic responses have been cloned in that position. That means to say that, see here what you're telling, your DNA is bringing about the changes in the embryogenic cells, how it has to develop along with your auxin, and along with your auxin, what even your environmental factors are playing the role. But whatever the role of the environment is playing, having influence on this, is a reversible change. Here the next sentence what is telling, see, your embryogenic development leads to changes in the expression. See, your embryo is going under various stages of changes, right? So when that it is going under various stages of changes, what happens is there is expression of a molecular marker that is indicating that, suppose say A simple example, just a wild example I'm giving. So this is your embryo. Now suddenly it has expressed itself. Okay. So here you can see this is your molecular marker. So when you see that, okay, it has increased or some change has happened, you come to know. So that will act as a molecular marker indicating that transition have been recognized. From what? From your somatic cells to somatic embryo, there has occurred transition, and there is a molecular marker to indicate that this type of change has been occurred in your embryo, I mean the somatic cells to embryogenesis formation. Also, the position in the callus and the appearance of the calcium in the vacuoles are the first sign of the embryogenic cells recognition. So, where is the callus? What is the position of the callus? And the appearance of the calcium in the vacuoles are the first sign of the embryogenic cell recognition. And also, students, another point is the cell wall associated proteins also participate in the embryogenesis. So you all know that the cell walls also have proteins, and they also participate in embryogenesis. It is very well documented that fresh culture can be induced by addition of cells free condition media. This is achieved by extracellular protein which is released by the embryogenic cells. So what it means to tell is, see, you have a cell, okay, and the cells will be having proteins, right? So the proteins will be secreted into the media. So then what happens? The cell wall associated proteins also participate in embryogenesis. So now you have the proteins which who has given these proteins? These are the cell wall proteins. Now where are these two present? They are present in your media. Now what you do is you remove off only the cells. Okay, from this media you remove only the cells and retain the proteins. So this is called as a cell-free condition media. This media, it is not having cell, but it is having the proteins. So it can grow well in such type of media. That is the meaning of this. Okay, cell wall associated protein also participate in the embryogenesis, and it is it can grow well in the presence of the cell-free condition media. Somatic embryos germinate immediately, resulting in the development of weak seed. Therefore, there is an additional state you have to take care of, because what happens is immediately the somatic embryos germinate, resulting in the formation of a weak seed. If the seed is weak, can we use it? No. Therefore, in order to have a healthy seed, you should make some alteration. That is, you should increase the gradual desiccation, increase the sucrose concentration, and abscisic acid treatment during maturation can help us to get a better cells or better survival of the seed. So these all precautions, whether it is epigenetic or your post translation modification or your protein protein interaction or the indication of the molecular markers which indicates that transition has happened, which tells you that from the somatic cells you have been developed into a somatic embryos, it is indicating us. And the callus position, the vacuoles containing calcium are all the signals for the formation of somatic embryogenesis. And also you can provide cell free condition media. And you know that the seeds, because the somatic embryos, it develops so fast, much of multiplication and differentiation and de-differentiation happens that it ends up in developing the seeds very fast, but the seeds are weak. So you should take care that these, instead of producing a weak seed, starting itself I require and healthy seeds. So you can gradually go for the desiccation, that is the drying. increasing the sucrose concentration and by abscisic treatment during maturation can help us to get a better the cell for the survival. So for germination, may obtain 3 to 5% of germination. Sucrose like 10%, mannitol for 4% may be required. So maturation of your somatic embryo means the four stages. Final stage is your shoot and the root formation. That is the cotyledonary stage. This embryo then starts to germinate. This one. It starts to germinate now. Germinate and it produces what? It produces a plantlet and young plant. Embryo germination, it may be necessary to chill the young or the mature embryos for the normal maturation development into plantlets. So sometimes this chilling of the young or young mature embryo is necessary for the development of the plantlets, and also application of your gibberellic acid, that is GA3, is also required for root and shoot development. Particularly, it is a very good example and in the citrus fruits. So this picture is taken from the Nature reviews. You can see the various stages of the development of the embryos, and you can see here that all what I had put it in a small form. Look at the genes which are playing the important roles, the different types of genes which are essential. And look at this embryo which is in the heart shape. You can see the shoot and you can see the roots stem cells. I told you, no, the cotyledons keep on uh, dividing during your heart shape, and finally in the cotyledonary stage we can clearly see the shoot and the root stem development. Okay, with this we can conclude today's class, and thank you for kindly listening to me.